All right, let's get straight into it. Today, we're tackling one of the biggest ideas in all of biology, variation and evolution. This is a core part of your AQA GCSE syllabus, so in the next few minutes, we're going to make sure you've got all the key concepts locked in, ready for that exam.
So here's our roadmap. We're kicking off with the absolute foundation, variation. Then, we'll see how nature uses that variation through natural selection. After that, we'll pivot to how we humans have taken the driver's seat with selective breeding. Then, up to the ante with genetic engineering, and finally, the most direct control of all, cloning. It's a really cool story of going from just watching nature to actively changing it.
So, to get us started, let's think about a massive, fundamental question that really drives this whole topic. Why isn't everything just a perfect copy of everything else? Why do we see all this incredible diversity of life on Earth?
Well, the answer starts with one simple but super important concept: variation. Think of it like the clay that evolution gets to sculpt with. Without it, nothing changes.
So what exactly is variation? It's nothing complicated, really. It's just all the differences you can see between individuals in any group. Just look at people or dogs or even pigeons in the park. No two are exactly the same. That's variation in action.
Right, so where do all these differences come from? Well, it boils down to two main things. First up, you've got genetic causes, the unique mix of genes you get from your parents. Then, there are environmental causes, things like diet or, for a plant, how much sun it gets. But honestly, for most things, like human height, it's actually a combination of both your genes and your environment working together.
But hang on, let's trace this back even further. Where does a new genetic trait come from in the first place? The ultimate source for all of it is mutation. These are just random changes that happen in an organism's DNA. Now, most of the time, they do absolutely nothing, but every once in a while, a mutation can create a totally new characteristic, adding fresh material to the variation pot. Okay, so we've established that there's all this variation out there. What happens next? This is where the engine of evolution really starts to roar. Natural selection. It's the process where nature itself selects which of those variations are the most successful.
Now, you absolutely need to know this four-step process for your exam. First, you start with variation. Because of this, there's a struggle for survival, a competition for resources. Some individuals happen to have traits that give them an edge. This makes them more likely to survive and reproduce. And because they reproduce, they pass on those successful genes. Do that over and over for generations, and the whole species begins to change.
So what's the grand finale of all this? Well, if you let this process run for millions and millions of years, and a population gets split up and isolated, natural selection can drive them in completely different directions. Eventually, they become so different that they can't breed with the original group anymore. And boom, that's speciation. A brand new species is born. So, that's how nature gets the job done. But what happens when we humans decide to take control of the process? Well, we've been doing it for thousands of years. It's called selective breeding. And you can think of it as natural selection, but on fast forward, with us making all the decisions. The textbook definition is pretty simple. It's when humans pick plants or animals with characteristics that we like and breed them together. Think about it. Creating crops that give more food, cows that produce more milk, or even dogs that are super friendly. It's all the same principle. We are the ones doing the selecting. This slide here really nails the key difference, and it's something examiners love to ask about. With natural selection, the pressure comes from the environment. It's a pure battle for survival. But with selective breeding, the pressure comes from humans. It's all about what we find useful or what we think looks good.
But, and this is a really big but, this process has a major drawback. When you keep breeding from a small group of related individuals, you get inbreeding. This shrinks the gene pool, which means there's less genetic variation. And the big danger there is that nasty, harmful genes can become more common, which can leave a whole breed susceptible to diseases and inherited problems.
Selective breeding is powerful, sure, but it's still pretty indirect. What if you could just skip the breeding part altogether and edit an organism's genes directly? Well, thanks to modern technology, we can. Welcome to the incredible world of genetic engineering.
So genetic engineering is all about directly changing an organism's entire genetic instruction book, its genome. The idea is you can take a useful gene from one organism, say the gene for making insulin from a human, and stick it into another organism, like a bacterium. And just like that, you've turned the bacteria into tiny little insulin factories.
Okay, for all you higher-tier students out there, you need to know the nuts and bolts of this. It's basically a three-step recipe. First, you use special enzymes like molecular scissors to isolate the gene you want. Second, you insert that gene into a vector, which is like a delivery vehicle. A common one is a little circle of DNA from a bacteria called a plasmid. And finally, you use that vector to transfer the gene into the cells you're targeting.
Now, as you can imagine, this technology stirs up a lot of debate, especially with GM crops. For your exam, you need to be able to see both sides of the argument. The potential benefits are huge. More food, crops that can fight off pests, even food with more vitamins. But the concerns are real too, like the impact on wildlife and questions about long-term effects on our health. It's a classic balancing act between progress and caution.
And that brings us to our final topic, which is pretty much the ultimate level of genetic control. We've gone from selecting traits to moving genes, and now we're talking about making an exact, genetically identical copy of an entire organism. This is cloning.
Now, there are a few different ways to clone something. With plants, it can be super simple, like when a gardener takes a cutting from one plant to grow a new one. That's a clone. But with animals, as you'd expect, the methods get a lot more complex, from splitting early embryos to the technique that made Dolly the sheep famous, adult cell cloning.
So let's quickly talk about that famous method. Think of it like a cut and paste job, but with cells. You take an egg cell and carefully remove its nucleus, its genetic information. Then you take the nucleus out of a normal body cell from the animal you want to clone, and you pop it into that empty egg. A little zap of electricity gets it to start dividing, and you've got yourself an embryo that's a perfect genetic copy.
And no surprise here, cloning is another one of those topics with huge arguments on both sides. The potential is mind-blowing. We could save endangered species or even grow new organs for people who need them. But the risks are just as big. Widespread cloning could crush the gene pool, making a species really vulnerable to a new disease. And of course, there are some very serious ethical and moral questions that come with it. So if we pull back and look at the whole story, it's pretty incredible. We've tracked this journey from simply observing the natural variation that exists all around us, all the way to a point where we have direct, hands-on control over the genetic code of life itself. We've learned the rules, and now we're starting to rewrite them.
And that leaves us with a really powerful final thought. The big question for us today is no longer, can we direct evolution? We're past that. We know we can. The real question now is, should we? And if we do, where in the world do we draw the line? That's everything for this quick run-through. Best of luck with your revision.